So now that we've looked at the structure of the glycerophospholipids and sphingomyelins, let's now look at how they come together to make a cell membrane. A cell membrane is a collection of these phospholipids that are arranged side by side They are, you can kind of see from this picture their arrangement side by side. The heads of the phospholipids line up side by side, head to head, and then the tails are lined up as well. So we've got a collection of phospholipids that are arranged side by side. The purpose of them, of the cell membrane, is to separate the contents or the inside of the cell from the exterior. So they hold everything inside the cell. As you can see from the picture, and as you probably already know from biology, the phospholipids come together in a double layer, which we call the lipid bilayer. And that is just simply a double layer of lipids heads all lined up, tails all lined up, and the tails of one layer lined up with the tails of the other layer. So let's go back to this diagram again. So we've already drawn in a little bit, the heads all lining up and the tails all lining up, and this is one layer of lipids. And then up here is our second layer of lipids with its heads lined up and tails lined up. And the tails of the two layers are in contact with each other. And if you think about that, it's going to make sense because these heads are polar. I almost wrote nonpolar. The heads are all polar, and these ones down here are polar as well. The tails are all nonpolar. So the tails of one layer are attracted to the tails of the other layer, and that's why they align together. The polar heads are attracted to water, aqueous stuff, which is what we have inside and outside of our cell. So the interior and exterior of our cells are in contact with the polar heads because they are compatible, and the tails, which are not attracted to the aqueous environment inside a human body. The tails are like isolated inside the middle of this layer. So this is, according to this diagram, this is the outside of the cell up here. This is the cells outside, and this is the cells inside. And this is kind of a tricky diagram, kind of a tricky drawing to look at. If we want to imagine, we have to remember that the cells are three-dimensional spheres, not flat layers. So if we were going to try to draw what this would look like, we would have to try to draw it in a circle, in a sphere, but I can't draw a sphere. And there would be like a bilayer of lipids in the sphere, sort of like this. And this would be all the tails of one layer, and here would be the tails of another layer. And this would be all the heads of one layer, and here's all the heads of another layer. And this is the inside of the cell, and this is the outside of the cell. And I'm just really bad at drawing, but that's kind of what it would look like. The purpose of a cell membrane, in addition to holding the insides of the cell in place, the function of the cell membrane is to move things in and out of the cell. Function of the cell membrane, in addition to holding stuff in, it moves things, molecules, ions, atoms, moves things in and out of cells. And there are 3 different ways that things get moved in and out of a cell. The easiest one to understand is called simple diffusion. And in simple diffusion, we are moving small molecules in and out of the cell. Small molecules that are moved across the cell are commonly oxygen, O2, carbon dioxide, CO2, and water, H2O. So we're moving these small molecules across the membrane, across, we'll call it the lipid bilayer. I don't think I got that one right. Lipid bilayer. Small molecules moving across the lipid bilayer. And they're moving due to a difference in concentration, meaning that we are getting a buildup of, let's say, oxygen, O2. We're getting a buildup of oxygen on one side of the cell. The concentration of oxygen is greater, let's say, on the outside than on the inside. So we have this difference in concentration, and this difference in concentration pushes the oxygen molecule across the cell membrane. In simple diffusion, our small molecules move from the high concentration area to the low concentration. And it's called simple diffusion because these little molecules simply just squish their way through the membrane like it's a piece of cake. And let's take a picture of, or take a look at a picture of what that looks like. So this first, on this drawing, this first representation right here is our simple diffusion, our picture of simple diffusion. And they're showing you that on the outside of the cell, there are 3 of this red molecule, whatever this red molecule is. And inside the cell, there's only 1. So we have 3 versus 1, which causes a concentration gradient. This is our high concentration side. This is our low concentration side. And so these red molecules just squeeze their way through into the inside with the goal of evening the concentration out so that the concentration is the same on both sides. Another way that we can move objects across a cell membrane is called facilitated transport. In facilitated transport, we are moving big molecules, specifically glucose, and we're also moving ions, typically chloride ion and HCO3 minus. This isn't where we move all of our ions, just some of them. We're moving big molecules and ions through proteins. We call protein channels. So these are like gaps or kind of like tubes inside the cell membrane that help move the big molecule and move these particular ions through the membrane. And this, again, is being moved due to difference in concentration. And also, just like simple diffusion, It is moving from the high concentration to the low concentration. And so let's take a look at a picture of facilitated transport. So that is being shown in this middle part of the cell membrane picture here. This thing in the middle is our protein. So this is the channel or the gap that helps move the molecule or the ion across the membrane. It's there because our big molecules like glucose just can't squeeze their way through, and ions like chloride and HCO3 minus, because they're ionic, they have a harder time working with the non-polar tails. It's just their polarity is too different. So again, we're going from an area with high concentration, 2 molecules in this representation, to a low concentration area. Down here we have 1 molecule, so high concentration to low concentration through these little channels. And the last type of transportation in and out of a cell is called active transport. Now, I've heard from students that biology, in the biology textbook that we use in 160, has different names for at least one of these 3 things. And I can't remember which one or ones are called by different names. And that shouldn't come as too much of a surprise to you, that we don't use all the exact same names. So if you're saying, hey, isn't that supposed to be blah, 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 whatever they call it in biology? Yeah, it probably is the same thing. So active transport is where we are moving ions, and this time we're moving hydrogen ions, H+, sodium ions, Na+, potassium, K+, calcium, and also chloride ions too. So they've got a couple options. Ions that are moving, again, they go through the protein channels, because their ionic nature is really repelled by the little tails. And with active transport, we are moving due to a difference in concentration, or against a difference in concentration. Against a difference in concentration. So this is a really different situation. This would be a situation where a cell needed something inside, and even though the concentration inside the cell was greater than the concentration outside of the cell, still needed to move that substance into the cell. So here we are moving from a low concentration to a high concentration. With active and facilitated transport, typically that situation is where we've got a buildup of stuff outside the cell, and we're moving stuff into the cell just to kind of equalize the concentrations in and out. With active transport, we have a buildup of stuff inside the cell, let's say sodium ions, but biologically or metabolically, we need more sodium ions to come into the cell, even though there aren't very many out here. And so we have to push, our body has to push those ions into the cell, even though Mother Nature doesn't really want it to go in. And so here is a representation of active transport. In this last picture, it looks very much like facilitated transport. It's using the protein channel, but here we are moving from the low area, which we have just 1 molecule here. We are moving from low to high. And when we're moving from low to high with 3 molecules, it takes energy. So energy is required for this process, whereas the other 2 processes just happen spontaneously.